Good morning. It's a pleasure to be with you today, and I want to say a big thanks to Dr. Carrillo for what a wonderful talk and for being with us. And a special big thank you to all the research volunteers here at University of Kentucky who are vital teammates and are incredibly important for all that we do. So thanks to all of them. This is the topic of my talk today. It's talking about late. It's a disease that causes dementia. And it sort of exists in a context. There's been recent increased appreciation of a complexity that Dr. Carrillo mentioned very nicely about the diseases that cause impaired thinking of memory and aging. The symptoms that we're talking about are memory problems, thinking problems, functional problems. These are called the dementia syndrome that Dr. Carrillo talked about. And what's really understood recently is that more than a single disease underlies those symptoms. So diseases including Alzheimer's and late, which I'm going to talk about, and many other diseases contribute to the dementia clinical syndrome that Dr. Carrillo was talking about. In order to understand that context, though, I have to give a quick primer about how the brain works. And it's going to be really fast because we've got a lot to talk about in a short amount of time. This will harken back to your high school biology class, where one of the functional units of the brain are neurons. We have 100 billion of them, but this is sort of represented in this cartoon here, this one neuron, and it works by communicating with other neurons, and this place that it interacts with is called a synapse. So this is a very functional unit of the nervous system. And to understand what's happening in Alzheimer's disease and other dementias, including late, you have to sort of understand proteins. So proteins are normally functional, and they help with both the structure of the brain, shown here on the left, and function of the brain. All those things are subserved by proteins. So normally you have these happy proteins, but for reasons that people don't completely understand all that well, the different proteins become dysfunctional in neurodegenerative disease. They become gloppy, they become misfolded. And so what's going on there, we are still working on understanding, but one thing that's known is it's not just one or two different proteins, but a number of different proteins. So in the course of dementia illnesses, that structure that was helped by the proteins, that becomes impaired. And the function also becomes impaired by these gloppy proteins. So getting back to our sort of thought of how the brain functions, it gets to a question of we call these neurodegenerative disease. So what is degenerating and why does it degenerate? Well, one of the big clues seems to be that within our nerve cells, there are these gloppy proteins build up. Also, in the structures that communicate between the nerve cells, including the synapse, there is an accumulation of these gloppy proteins. So this gets to our point of understanding and differentiating between the different conditions that we see that contribute to the dementia syndrome. So, for example, Alzheimer's disease, that Dr. Carrillo was talking about, is contributed to by misfolding glop in the tau protein and the A beta peptide. Whereas Parkinson's disease and dementia with Lewy bodies are usually due to a separate misfolded protein called synuclein, and late, as well as Lou Gehrig's disease, which is also called ALS, and other diseases that are sort of a grab bag that are called frontotemporal dementia, can be contributed to by a separate protein called TDP-43. And that is one of the things we're talking about, is how late and TDP-43 are relevant to our story of dementia and how we're going to eventually address dementia illnesses. So late means literally, it's an acronym for limbic predominant age-related TDP-43 encephalopathy, which is a mouthful. It basically means in English, an old person's dementia disease with misfolded gloppy TDP-43, but we have to use long words to describe that. Late, the implication is that it actually preferentially affects people that are late in the human aging spectrum. So this is one of the characteristic things we see under the microscope with TDP-43. You see these inclusions inside of the nerve cells, But then you see these other diseases with A, beta, and tau. This is Alzheimer's disease when we see these under the microscope. Or these are special stains looking under the microscope for alpha-synuclein when we see in Parkinson's. It's classically thought that these diseases segregate into these specific diseases, but it's more and more increasingly seen, as Dr. Carrillo mentioned, that they're often found in mixed and combinatorial patterns. And this is from a paper that's been cited well over 1,000 times. It was a consensus paper. But one thing that our University of Kentucky friends should know, that all these pictures were taken right here at the University of Kentucky, testifying to the fact that we have had an impact on this field. So these things are not, though, just academic issues. They're the diseases that affect people. They affect all the populations. It is a huge impact on so many people. It deprives people of their memory, of their spirit, of their ability to function. It just can't be understated, the impact on society, but also affects, of course, individuals. So I'm going to present a few individuals from our cohort. This is the first person is a person that enrolled at age 77 and then was followed for 25 consecutive visits. This is the mini mental state examination test score. This is 0 to 30 test that many of you have taken as part of our studies and have annoyed you for years, but they actually come in very handy to document the trajectory of the impairment of this person. At age 91, she was diagnosed with mild cognitive impairment, and by age 96, she had dementia that was thought to be Alzheimer's disease. One thing that I just want to underscore is that these are incredibly powerful data that you can use, and I hope to convey to you, at least give some glimpse of that in a later slide of some of the ways that we've used these data. But this particular person had no Alzheimer's type changes, no plaques at all, but had late instead, TDP43 proteinopathy. So this is case number 1. There was a GLP protein. It was TDP43 looked at under the microscope, and that's all that she had. Here's a second person that was also enrolled at the University of Kentucky at the age of 65. This person was followed for 27 years and was diagnosed with Alzheimer's disease at age 84, and it had a very bad rocky road after that. This person had a combination, actually, both severe Alzheimer's disease and late. These are both very common pathologies. So in this case, there was 3 different misfolded proteins, tau, A beta, and TDP43 proteinopathy, all visible under the microscope and diagnosable. And this is an incredibly common disorder. So who cares? This all seems sort of academic, pin of a head, how many angels, blah, blah, blah. But it's actually very important. This shows data from a cohort that Dr. Carrillo mentioned, the Rush University community-based cohort, one of the better ones in the world. And it shows the prevalence in different age ranges. So this is 75, 80, 90, and over 100. This is proportion of people with a pathology. These are the people with severe Alzheimer's disease. And one of the things you can see is that the proportion of people with severe Alzheimer's disease goes up, but then sort of comes down in a very, very advanced age. By contrast, late, this is late in the human aging spectrum, keeps on going up and up and up. Late affects around 1/3 to 1/2 of people over age 80 in cohorts all around the world. And so this is a growing appreciation of the impact of this pathology, which is right up there with Alzheimer's and other neurodegenerative and cerebrovascular conditions. So some of the workers in our own center that I'd like to hype a little bit, Shama Karanth, who is a wonderful grad student, and her boss, Dr. Aaron Abner, who was the spearhead of this study. And this is a nice study that is hot off the presses in a good journal, JAMA Neurology, here in 2020, looking at the proportion of people with dementia from our cohort that have been followed for a very long time. And 46.3 of them have that combination of late and Alzheimer's disease, whereas only 19.2% have pure AD. So that means it's more than twice as likely to have this combination than that which people around the world had been associating with disease for years and years and years. And so again, so what? Who cares if there's another pathology there? Well, it turns out it really matters. And this is one of the applications of those great data that I showed you before, when you have hundreds of people that you follow to autopsy, having had many, many longitudinal studies. And so again, we just thank so much. We're so thankful for our research volunteers for going through this year after year after year, because it's extremely powerful and helpful to have those data. And here's one example. This is showing the, again, this is Shama Karanth and Dr. Abner's work that was recently published, showing the trajectory of cognitive impairment before these people were dead, before they passed away. And it shows that pure late has a relatively mild decrease of cognition. Pure AD is somewhat worse. But the people that have, and this is two groups of people that have both AD and late, they are still worse. So there's an additive impact of this very common set of pathologies that makes it worse to have both of the pathologies. So it turns out that what we have for dozens and even 100 years or so thought was Alzheimer's disease contributing to dementia, now that we can look at it, we understand that the actual contribution of Alzheimer's per se is more mild, whereas the combination is what contributes within an individual. This is cognitive changes over time, that it's the combination that really drives a worsening prognosis. And many people, and a lot of people, just have late. So in a way, this is a bummer. Why? Because it says that not just one thing can get you when you get old and cause you to have a dementia clinical syndrome, but other things can too. So just because you've survived this burning building and you're rescued, well, now you can bounce over here and hit another pathology, which is called late. And so it's kind of a bummer that multiple things can afflict us as we get older. But to me, it's actually extremely exciting. It's extremely exciting because the different subtypes of dementia are each a therapeutic target in themselves. And it's not until we grapple with that complexity that we have a chance at curing everything, including the Alzheimer's in the first place. So with that in mind, to discover disease mechanisms for therapeutic targets, because we're always focused primarily on preventing or treating the diseases, that we're not here just doing work to do work. We're doing work to try to cure diseases. We ask genetic risk factors because that can give you a handle that enables you to perhaps have an opportunity for a therapeutic strategy. We did the first genetic study of late. Right here at the University of Kentucky. The person who sort of ran the show was Dr. Dave Fardo, who is a statistical geneticist, and he did great work. And he found that there's an association between genetic variation in a gene called ABCC9 and late. I'm not going to get into all the interesting aspects of this because I'm going to really focus here on what we are primarily focused on ourselves, which is trying to accomplish one of the great unachieved goals of genetic studies, which is clinical relevance. And the reason for that is that the gene that we found associated with late, which is called ABCC9, is in fact a potentially druggable target. So remember, a gene codes for a protein. This protein is kind of a little tube. It's actually kind of small in real life, and it's integrated into a biochemical context. So this is the surface of a cell. The tube is inserted in that actual membrane of the cell. It acts as a channel, and when it's opened, it produces a cascade of biochemical events that leads to a more healthy environment inside the cell. So I'm not going to get into the weeds here talking about exactly what it does, but it leads to a more healthy environment in the cell when that channel is open. And there's a drug called nicorandil, which is used in countries around the world to treat old people with congestive heart failure. And so this drug acts by potentiating or causing the opening of this channel, which leads to a more healthy internal environment in the cells. So our colleague and clinical core director, who we're going to be talking to shortly, Dr. Greg Jicha and myself are now funded for a project to assess the impact of this drug in a randomized, placebo-controlled clinical trial to see if people at risk for late can be treated with nicorandil. This involves a very complicated clinical activities, which are shown here on this very complicated table that Dr. Jicha made, all sorts of tests and counts and adverse events assessment and screening at various times. But I think it's an extremely exciting thing. Dr. Carrillo mentioned that there were, when they did the PET scans, there was a finding that so many of their patients were negative for Alzheimer's, even though they had cognitive impairment. A lot of those people are going to be at risk for late. And so I think that is a part of a very exciting story about this newly appreciated disease called late, which is limbic predominant age-related TDP-43 encephalopathy. It's important because it's extremely common. It's now appreciated to be a third to a half of people in advanced old age, and it mimics and worsens Alzheimer's disease clinically. And we might beat it using the very first late clinical trial that is based upon the very first genomic screen that was all performed right here at the University of Kentucky. So there are too many people to thank and too little bit of time to thank them, but the team here at University of Kentucky is so amazing, and it's just something I thank myself every day to be able to work with these wonderful people, and in particular Dr. Van Eldik, who is an outstanding leader, and I think it's really always very important to choose your boss wisely, and she's been a wonderful boss. So with that, I will say thank you.